All right. So I wanted to talk to you about a plate count experiment and why we do them, and most importantly, how we do the math that's required to get results from a plate count experiment. I wanted to start out with some information from a page in your course pack, which introduces the issue of counting bacteria in a sample. And that's page 54 in your course pack. And I've got some of the text from it right here. We don't usually cover chapter 6 in your lecture textbook during lecture because we work with growing microorganisms in lab so much. This semester, I would recommend that you read the last part of chapter 6, though, the part that talks about measuring microbial numbers and specifically about plate counts. What you'll see, if you read that, it's the last part of chapter 6. You'll see that we often do want to know how many bacteria are in a sample, and there are several ways that we can measure them. One thing that will not work to measure bacteria in a sample is a streak plate. It just won't give you the information about the concentration of bacteria. So three common approaches that are described in the reading are listed here in red. And probably the simplest one is just to take a sample and put it on a microscope slide and count how many cells are in the liquid that's on the microscope slide. That's a direct count. An indirect method we can use is to measure how cloudy a liquid culture is. That cloudiness is called turbidity. And the third method, the one that you will do in lab, is a plate count, or a viable or standard plate count. Each approach has its advantages, and the cell count, the biggest advantage is all you need is a microscope, a way to measure your sample, and a person who can use a microscope. So it's very low tech, you get results pretty fast, and it's easy to do. Turbidity is a little bit more of an outlay in terms of the costs of the equipment. You need a special piece of equipment to measure turbidity, and you need someone who knows how to operate it. That being said, turbidity can be a good choice for estimating microbial numbers because with a good spectrophotometer, that's the equipment you use, you can check thousands of samples, literally thousands of samples a day, if you have a lot of water samples from, say, different parts of a municipal water system. The plate count is the one that takes the longest and it uses the most material. But the big advantage with a plate count is that it tells you what you usually want to know, which is the number of live cells in your sample. A cell count or a turbidity measurement will not normally distinguish between live and dead cells. And we care a lot more about the live salmonella in a sample than the dead ones. So you're going to be doing a plate count experiment, and it's going to look similar to the one that's shown in your book in figure 6.16. And I'll put that up in a minute and walk you through it. But on the bottom of page 54, it gives you a place where you'll be able to do the calculations once you get the results from your plate count. And one thing that's shown here is that we're going to be looking for a plate in the plate count experiment that has a certain range of colonies on it. We need a plate with between 30 and 300 colonies. And that's the only plate we're going to care about in our results. And we need to know the dilution factor of that plate. And once we know those two things, it's a pretty simple calculation to determine the concentration of bacteria that were in the original sample in the experiment. And that's always what you're trying to determine. You take the number of colonies on the countable plate, divide by the dilution factor of the plate, and that is your original concentration of cells in the starting sample. And that's expressed in units that are often referred to as CFUs per milliliter, colony forming units per milliliter. It corresponds more or less to live cells per milliliter, but it's a little more accurate to refer to them as CFU. So that's why you'll often see your book, for example, refer to this number as CFUs. All right, so let me take you to the figure from your textbook, figure 616. It is on page 167 in your book if you want to look at it there. But this, I'm showing it to you on the screen now. So it starts with a serial dilution that's done in tubes. And in this case, each tube gets 1 milliliter of the previous container added to 9 milliliters of sterile liquid. This will create a series that we refer to as a 10-fold serial dilution. If you look at the dilution factors, which are written underneath the plates in this experiment, you can see that they range from 10 to the minus 1 to 10 to the minus 2 to 10 to the minus 3 to 10 to the minus 4 to 10 to the minus 5. So the dilution factor goes down by 1 power of 10 with each tube in the series. After you make this series of tubes, you take 1 milliliter from each tube, spread it onto an agar plate, and then incubate the plates for several days. You come back and look at the plates, and they have a range of colonies on them. The plates with the lower dilution factors, 10 to the minus 1, 10 to the minus 2, have a lot of colonies. Whereas the ones with the lowest dilution factors, 10 to the minus 4, 10 to the minus 5, those last plates have few colonies. The assumption is that each live cell that's put on the plate forms a colony. So if we know how much of the original sample made it onto each plate, and we know how many colonies are on the plate, we can calculate backwards to get an estimate of the number of live cells per milliliter in the original stuff. That's the CFUs per milliliter. Okay, so the first step with a plate count after you have the plates as your result is to figure out which plate to use for the calculation. So remember what it said on page 54 is we need plates between 30 and 300 colonies. If it's fewer than 30, it's not an accurate snapshot of what was in the original. It's just too diluted. If it's 300 colonies or more, it's going to be real hard to calculate or to count it accurately. So the plates that are over 300 colonies, you just can't count them accurately. And we usually mark them or record them as TNTC, or too numerous to count. We never would use a TNTC plate to calculate results for a plate count experiment. The plates at the very lowest concentrations give us 16 and 3 colonies. Seems like maybe you could use that 16 colony plate to do a calculation, but the problem is it's under our 30 colony minimum. So we really have to use the plate in the middle. In a plate count experiment, there will be a single plate that is the right plate to use for the calculation. And here it's the one in the middle. So the caption for this plate in your book said that plate has 54 colonies. It has more than that, I'm pretty confident. My best count of the plate gave me 154 colonies, which seems about right because these are 10-fold dilution. So each plate should have about 10 times more colonies than the next plate in the series. And since the next to the last plate is 16 colonies, we'd expect that middle plate to have about 10 times that, or 160. So let's go with 154 as our colony count on that middle plate, and that is our countable plate we can use for the calculation. Underneath the plate, we see the dilution of this plate is listed as 10 to the negative third. So what we need to do is we need to take the number of colonies, which is 154, divide by the dilution of the plate, the dilution factor. So that's 154 divided by 10 to the negative third power. I'll remind you that when you're dividing by a power of 10, you can bring the number up to the top of the fraction by changing the sign of the exponent from positive to negative or negative to positive. So our number on the bottom of the fraction here is 10 to the negative third power. We can bring it to the top and rewrite that as 154 times 10 to the third. That's our CFUs per milliliter. And if you get that far, if you get as far as this, 154 times 10 to the third, on the worksheet or on the lab exam question about a plate count, I would give you full credit for it. If you do scientific notation in other classes, you may know that it is not exactly correct scientific notation to write 154 times 10 to the third power. That first number and the number that's written in scientific notation, the first number, which is called a coefficient, is really supposed to be between 1 and 10. So if you want to move it into completely correct scientific notation, you would take 154, rewrite that as a little more than 100. So 1.54 times 100. 100 is the same as 10 to the second power. So then you have 1.54 times 10 to the second power times 10 to the third power from the dilution of the plate. And you can add those exponents together from the 2 powers of 10 to multiply 10 to the 2 times 10 to the 3. You add the 2 and 3 together. And so the other way that you could write your answer is 1.54 times 10 to the 5th CFUs per milliliter. If you have either of those two, either of these two as your answer, I'll be perfectly happy on a worksheet or on a lab exam in this class. We're going to make sure that you can in fact do these calculations. So in addition to calculating your results from your actual experiment that you do in lab, I'm going to have you try some practice questions, which are on page 59 in your course pack. And you'll turn these in along with your dilution series practice. So let me just talk you through what is on page 59. What we have on page 59 is actually 4 separate experiments. A, B, C, and D are each a different plate count experiment. Each experiment has 4 plates in it, and for each plate, the dilution of the plate is written above the number of colonies. So in our first experiment, we have a 10 to the minus 2 dilution factor, 10 to the minus 3, 10 to the minus 4, and 10 to the minus 5. Those are the 4 plates. The first 2 plates are TNTC. The third plate has 142 colonies. The last plate has 10 colonies. So the first step with any of these, either on the worksheet or on the lab exam, the first step is picking your plate to use for the calculation. So remember, you want to use the 30 to 300 range plate, and you'll take the colonies on that plate and divide by the dilution of the plate. Convert that number into something that looks like scientific notation. So in other words, a number times 10 to the something. I don't care if your first number, the coefficient, the number is over 10, that's okay with me, but it should be in the general form of scientific notation. Something times 10 to the something CFUs per milliliter. There will only be 1 plate from each experiment that is the correct one to use for the calculation. So experiment A will only have 1 answer. Experiment B will only have 1 answer. Experiment C will only have 1 answer. And experiment D will only have 1 answer. You do 1 calculation for each experiment. And if you can do the plate count examples on this page, page 59, you will be able to do the plate count question we ask you on the lab exam. There will be 1 dilution series where you need to make calculations of the dilutions of the tubes. And then there will be a separate question where the dilutions will be given to you. The colony counts will be given to you just like here on page 59, but you have to do the calculation to come up with CFUs per milliliter.